Today we can discuss about haptens. What are haptens? Haptens are small organic molecule which are antigenic but not immunogenic. These molecules are antigenic but they can't be immunogenic. They can't activate B or T cells. For activating B cell or T cell, hapten need to combine with a large carrier protein. Then only haptens can be immunogenic. They can activate the B cell or T cell then only. We can see in this figure the hapten when binds to the B cell, it can binds to the B cell but it can't activate the B cell to and produce antibodies. So it binds to the B cell but it can't activate it and produce the antibodies. But if the hapten carrier conjugate is there, it can activate the B cell as well as the Th cells. So we can see in this figure haptens, a large number of identical haptens are there. When we inject that haptens to our body, it can't activate the B cell. But if these identical haptens are combined together with some carrier protein, it can activate the B cell and can produce the antibodies against the haptens. So these hapten carrier conjugate, when they are activating the B cell, they will produce the antibody, three types of antibody, usually three types of antibody. One antibody will be against a carrier protein, here it is the BSA, one antibody against hapten, other antibody against the junction or the new epitope formed between the junction of interaction between the hapten and as well as the carrier protein. So the concept of hapten came more strong by the work of Karl Landsteiner during 1920s to 30s. Landsteiner used haptens for finding the antigen antibody or for in the studies of antigen antibody specificity. So Landsteiner employed hapten, the small organic molecule that are antigenic but immunogenic for its specific for finding the antigen antibody specificity, because if we use some proteins or nucleic acid to find the antigen antibody specificity, manipulating these big macromolecules are very difficult. But if we take a small molecules, small molecules, it is very easy to manipulate. So he used these haptens for a haptens as a tool in studying the antigen antibody interactions. So, as we said earlier, he combined the hapten with the carrier protein and injected in rabbit. And as he injected in rabbit, we can see in this figure, he injected it in rabbit. So he injected only hapten to the rabbit, so no antibodies was produced. When he injected the carrier molecule, there is antibodies against the carrier molecule was produced. When he injected the hapten carrier conjugate, he got the antibodies raised against the haptens carrier conjugate as well as the junction between the hapten and carrier conjugate. So one of the majorly used haptens by him was the dinitrophenol. So he combined dinitrophenol with the carrier protein, that is a BSA, so that he found out that three types of antibody were produced in the rabbit. One is the antibody against hapten, that is the antibody against the dinitrophenol, antibody against the carrier protein, antibody against the BSA, or antibody to the conjugate of hapten and carrier conjugate antibody against the junction of interaction between DNP and BSA he got. So the beauty of the hapten carrier system is to provide immunologists to chemically define determinant that can be subtly modified by chemical means to determine the various effect of chemical structure in immune system. That is the main advantage. In the case of haptens, they are small organic molecules, so they can be chemically modified. But if you take a protein or a nucleic acid for studying the antigen antibody specificity, it is too difficult to modify them. But the haptens are small molecules; they can be chemically defined in various ways, so that they can be used for studying the antigen antibody specificity. So that is how Landsteiner studied. We have already discussed this PPTs. And Landsteiner tested, this was what Landsteiner done, tested whether an anti-hapten antibody could bind to other hapten that have slightly different chemical structure. Landsteiner take an hapten carrier conjugate and inject to rabbit. Then some anti-hapten antibody will be produced in the rabbit. then he is using chemical method, he slightly modify that hapten, and then he combine that hapten with another carrier protein. So now we have a new hapten, the chemically modified hapten with a carrier protein. He take it and inject to the rabbit, so that he then check whether cross reaction occur, whether this anti-hapten antibody can bind to the new chemically modified hapten which we have injected now. If it bound to the anti-hapten antibody bound to the chemically modified hapten, then the process is known as cross reaction. Then he studied that why it bind, what type of chemical modification made the cross reaction occur. If it doesn't if the cross reaction doesn't occur, he studied why the cross reaction doesn't occur, what type of chemical modification prevent the cross reaction. That in two ways he studied, and he came to a conclusion which type of chemical modification or which type of modifications results in the cross reaction or resulting in the antigen antibody specificity. So he also done his experiment with another hapten, that is a O-aminobenzoic acid, aminobenzene and its derivative O-aminobenzoic acid, M-aminobenzoic acid and P-aminobenzoic acid. He used another hapten, that is a aminobenzene, and he found out that the conclusion was the overall configuration of the hapten plays a major role in determining whether it can react with the given antibody. If the configuration of the hapten remains same, he had made the configuration of aminobenzene same, and if the configuration of the hapten remains same, the cross reactivity will not occur. The aminobenzene, the anti-aminobenzene antibody against aminobenzene will bind only to aminobenzene, or antibody against O-aminobenzoic acid will only bind to O-aminobenzoic acid. So if the overall configuration of the hapten remains same, the antibody will bind only to the original immunizing hapten. It will not cross react. But if maintaining the overall configuration of the hapten, we just modify the para position of that hapten with some non-ionic derivative. If some small modification we can do it in the para position while maintaining the original configuration, it was shown that the Antisera showed cross reactivity. If we maintain the overall configuration is kept same, and if we modify this, the specificity lies on the overall configuration. If we change the overall configuration, if we change the overall configuration, the cross reaction will not occur. While if we are using one hapten, DNP one hapten, and if we modify DNP at its para position without touching the overall configuration, if we inject that modified DNP also, the antibody which is produced against the DNP first will cross react with the modified DNP which we inject now. So cross reactivity can occur if we maintain the overall configuration same but modify at the para position. So Landsteiner work not only demonstrate the specificity of immune system, but also demonstrate the enormous diversity of epitopes that immune system capable of recognizing. So by this work, Landsteiner has demonstrated the specificity of antigen antibody interactions. Many biologically important substances such as drugs, peptide hormones, steroid hormones can access haptens. This is the major applications of haptens. This is the major role of applications of haptens, because drugs, peptide hormones, steroid hormones can access haptens. If they are combined with some carrier protein, and if we can generate the antibody against drugs, peptide hormones, steroid hormone, we can generate assay for detecting this peptide hormones, steroid hormones or drugs in our blood or in our system during any diseases. So this is the main advantage of these haptens. The conjugates of haptens, these haptens, drugs, peptide hormones, steroid hormones with large protein carriers can be used to produce hapten specific antibodies. These antibodies are useful in, as measuring the presence of various substances in the body, measuring the presence of drugs in our body during when we are treating some dreadful diseases. We need to check the presence of drug every time. Usually during this transplantation, etc. In order to prevent the rejection of organ, etc. So in that cases we need to check the presence of drug continuously. So in that case, if you have the antibody test available, it is very easy to test how much amount of drug is present in our blood, or how much amount of peptide hormone, or how much amount of steroid hormone. Mainly in the case of reproductive biology, when we are researching the amount of steroid hormones at particular stage of the reproduction is very important. So the measuring this steroid assays steroid hormone, etc., can be done using this this Haptens. So the original home pregnancy kit is also based on this Haptens. The original home pregnancy kit contain anti-Hapten antibodies to determine whether women's urine contain human chorionic gonadotropin. It's a sign of pregnancy. The original human pregnancy kit also used this Hapten as one of the use anti-Hapten antibodies for determining the presence of HCG in the urine of the individual. Sometimes this drug protein conjugate, drug carrier conjugate can lead to drug allergies. It may be life threatening also, so you need to be cautious while dealing with Hapten carrier conjugates. So some of the most widely used Haptens are dinitrophenol, aminobenzoic. We have already seen all this. O-aminobenzoic acid, M-aminobenzoic acid, P-aminobenzoic acid. Many biologically important substances which can function as Haptens are the drugs such as penicillin, peptide hormones, steroid hormones can act as Haptens. We can now see how Haptens can be used in pregnancy kit. The pregnancy kit mainly consists of two components. One is the Hapten carrier conjugate and anti-HCG antibody. We will take the urine and add the anti-HCG antibody and incubate it for some time, and then we will add HCG carrier conjugate. And if you observe visible clumping, it's a negative reaction, that is the person is not pregnant. How it is become negative? Usually the Hapten carrier conjugate and the anti-HCG antibody will cross react each other and form a clump. While if the person's urine has the HCG, the HCG will bind to the HCG antibody, and so that there will not be any anti-HCG antibody to form this clump. So there will not be any clumping. If there is no visible clumping, the test is positive. That is that much various applications in immunology this Haptens has. So hope you enjoy my lecture. If you enjoy my lecture, please subscribe to my channel and keep watching. Thank you.